Thank you. I'm particularly, I guess, honored really that the people doing something completely different would come and listen to me. So I hope it's not too boring. Thank you for giving me your time. So having said that, I also want to, let me actually first, I want to acknowledge, so experimental biologists tend to have large groups. I don't know if this is necessary or not, but somehow it seems to be the mode of the operation. I'm very lucky to have a bunch of, this includes Harvard undergraduates, graduate students, postdocs mainly, and so it's, I'm very lucky to have a good group of people. But what I was going to say is after this that the talk I've organized with actually quite a more than usual introduction, in part because I wanted to just tell you, yeah, maybe just the front lights would be good, thank you. Partly to just maybe I'm trying to sell, like, why is it interesting? Like, why would I bother studying this? Why is it interesting? And in particular, I'm hoping that I will touch something that would maybe excite people who are thinking more theory and computation. I think that's sort of the goal, because I think we all, everybody wants to understand the brain, is interested in understanding it at sort of this conceptual, I don't know if you want to call it theoretical or not, but how do you explain things? We're extremely far from it, but that's the motivation. So it's a relatively long introduction, and then the actual results from my group are relatively modular, so I can stop, so you can ask me any questions, even for the simplest things, and I can stop at any point, and it doesn't matter, because these are just stories, they're very interesting to me, but maybe not so important to you. So please feel free to stop me anytime. So just a point, the starting point I have this slide is to say is that a huge amount of animal kingdom, obviously starting at single cells, they sense chemicals, but if you now go to kind of bigger animals, the ones that use odors to actually guide their behaviors, right? I mean, they use chemical signals to guide their behaviors. There are, of course, other signals in the world that they use. Vision, I think a lot of people study. Sound, audition, a lot of people study. And chemical senses is actually quite important for certain creatures. I mean, vision is important for others. So the three examples I've chosen are somewhat deliberate. One, this is a crustacean sitting in water, so in certain kinds of fluid, and it uses what's antennule, it's kind of hard to see in this contrast, but little device that it can flick around, and then so this chemical world, and it's basically feeling the chemical world, and it uses this to find food, right? Not surprisingly. This is actually
what's the difference between an antenna and an antennule?
It's all terminology. Biology is, like, you guys have your symbols. Biology, we have our just words. So it's antennule is probably because there's a main antenna, and then this is probably coming off of that. I actually don't know, but I suspect that's what it is, yeah. So this is a certain kind of moth that in the dark, so without using vision, mostly our very dull vision, finds a particular flower that it likes to get nectar. So it's basically a source of food, and then it does that from this, flower is emanating all kinds of, beautiful fragrance, smells, and this moth is finding that from a long time, and there's some beautiful work done by insect people, particularly in Arizona, that specific species of moth are going to specific datura flower and so on. So this is very interesting. And a matching of these things. So they use chemicals. Right? This is now in the air, kind of long distance, lots of fluctuating stuff going on. Okay? Then this one, an example I just recently became aware of, that birds, typically people think, olfaction, they don't seem to have much of a nose and chemical smells, and they're really good vision and whatnot. This particular kind of vulture called turkey vulture apparently has a much more well-developed, not apparently, it does have a much more well-developed olfactory system with a very big olfactory bulb, the part of the brain that processes the early olfactory signals, big nostrils that are there, and there are apparently field experiments to show that they use those chemical sense to find the carrion. And in their particular place where they do this is very thick tree canopy, so vision is hard. Right? You can't really see it. Like if it's open, you just vultures, that's what you see. But here they are much better at it because they use, and this is also quite interesting because this is a very big scale here, and how this works is just, I think it's really a mystery, right? Which brings me to the point of, I think, in talking to, particularly my collaborator and colleague, Massimo Vergasola, among others, that thinking about the physics of actual landscape of what happens here is obviously important, right? It's just as biologists, we didn't always think. So if you're looking at very small spatial scales or very viscous things, then it's more or less just diffusion. Like a bacteria is just doing things by just looking at diffusion. Right? I mean, if it's a source of chemical, you're just mainly looking at, that's what, that's low Reynolds number. So around medium, it's really kind of the advex, the fluid carrying the stuff, right? I mean, it's relatively quote unquote smooth, but, and this is where that crustacean that I showed you, the thinking is that's the regime under which it's doing. So things are sort of flowing and they're doing. And then you have a relatively high Reynolds number where things are really fluctuating and turbulent. And the main point is that the directionality of things is actually lost quite a bit. And your encounters of the actual signal is intermittent, so you have to be much more clever, I think, to figure things out, because here you can do either a spatial gradient or a temporal gradient. Bacteria do temporal gradient, as it turns out. And then here too, I think, you can do some sort of time averaging, look, keep listening, and then things are continuously changing. Here, it seems like it's a different regime. And I think this is illustration actually is from, I think the experiments were from Emmanuel Villermoz and Antonio Solani and Bergasola were in this paper where they visualized jet flow, and you can see this fluorescent dye. I think they didn't put the spatial scale because it's all sort of relative to this and the flow, but very quickly, probably in real world in air, even less than a meter, things become extremely intermittent here. So if you sit in one place, if you are an animal trying to find this chemical that you need to go towards, let's say because it's food, if you're sitting close by, your encounters in time are going to be quite frequent. It's fluctuating, but you can kind of live with it and do some averaging. But here, you're sitting here and your encounters are very intermittent. And I think there was actually a formal method that one can use to navigate up this, essentially it's not a gradient, up this kind of field. And this is this paper that Massimo and Boris Schreiman had called infotaxis, right? I mean, basically, you can go up an information gradient. You can't go up a chemical gradient because there is no smooth chemical gradient. So, but they have this mathematical formalism where you can actually go up an information gradient, and maybe you can design a robot to do this. It's unclear if animals do this, but you have to be more clever. And the honest answer is we don't know exactly how animals go up this kind of a very difficult environment, and that's why this turkey vulture thing is interesting. If it is true that they're flying and they're really cleverly going towards higher concentration and finding the carrion, then how are they doing that? I don't know the answer to any of this, so this is not what I will be telling you, but this is one place where it seems like some combination of sort of physics and trying to understand what the goal of the organism is could be an interesting point. What we typically do now in my lab and other labs for any sort of this odor-guided behavior in naturalistic condition is to use surface-bound chemicals. I mean, that seems like a little bit more tractable because ideally, it's surface bound, so you can look at the extent of the signal, and then you can watch the animal actually follow the gradient. We had a paper that started as a hobby in the lab, and now it's becoming a little bit more, that everybody knows ants track pheromone trails. That's why they, you see them all, and they find the food. But it turns out that there's not an actual algorithmic description of how they do it. They have two antennae. Like how are they doing it? Are they measuring gradients across the antenna? Is that as simple as that? But maybe not, because we don't know the extent of the thing. Is it thick, thin, the trail? So that's not quite known. So what we instead decided to do was to actually paint in the lab a known pheromone trail, right? That's this ink. And we added the ink for our camera to visualize. The ants can't see it because the experiment is done in infrared light, so they don't see the thing, but the camera can see it. And if I can play that video, I don't trust this thing, so I'm going to play it here. She tracks it very beautifully. The two marks are actually marks determined by computer vision after the video, so that's not marked on the ant, right? This is a post hoc marked. And let me just play it one more time. So this is a relatively simple trail, right? Thin one, it goes straight and it takes, and it's interesting, there's not that much of an overshoot, right? I mean, it goes and then it quickly does it. So anyway, it seems like a modestly interesting problem, and we're actually, again, working with Massimo to see if there's some sort of a formal agent-based, this is how normatively the animal should be doing it. Like, for example, if you go straight and if you suddenly make a curve, like what should it do? Does it have a memory of the straightness and then curve? How soon does it realize, and so on? Yeah, question. Oh, excellent question. So this trail, it's ground up abdomen glands. So the ants, the way they go forage, when they're foraging, when they're finding, there's no trail because there's no directionality. Once they find the food, then they come back to the nest and then they put a trail because then it's more direct pathway to direct the next set of ants to go over there, right? So that they do by putting the abdomen down. It's the trail pheromone that they deposit along the way. The papers, people have studied ants for a long time. These are carpenter ants, by the way, to say. These are extremely common in the US in many different, I don't actually know if they're also similar in India, but these are these black ants which chew up wood, right? So we know in many species, people have described what the actual chemicals are that they follow. We tried getting those chemicals putting, it really didn't work. So we just decided we'll just take the entire gland, which is good and bad, it's easy. But it's bad because the glands have many different things, right? There could be alarm pheromone, trail pheromone. So we're empirically lucky that they seem to be okay and track that. So, okay, so that's one, there's some interesting behavioral things to do. And can we do the same kind of thing in mice, which is almost all the work in my lab is in mice, partly for historical reasons and, there's just the weight of all the neurobiology on this animal. They're obviously very olfactory animals, so they can track. So what we did was that I won't talk about it because this project is not quite mature, that we developed, so the ideal thing would be to, again, draw some trail for the mouse and watch the mouse. But the spatial scales are too much, right? I mean, they run really fast. How do I put a camera? How do I get a big arena? So what we did was we took an inkjet printer and put it on an infinite paper treadmill. So we made a treadmill. So the treadmill is continuously going and the inkjet printer is basically printing the trail, which has some smell, right? And they're just curious about the smell, and then they would track it. So that's why this treadmill is going continuously, and then we're watching that animal in a small enclosure and that.
Are they selective about the smell?
They are, but not in this experiment. We haven't tested and that, but in general, if you do it, but you could do all kinds of experiments. So here, the goal is that if we have this working well so that we have large parts of the time that they're tracking, we can have the geometry. This geometry of the trail, by the way, was done by more physicsy person. They wanted to simulate a levee flight. I have no idea why, but I think it was probably not the right. It should have been much more smooth trail. That would have been much easier to follow. So we do more smooth trails. Once we do that, then you can give choices. Right? You can say, and then you can, for example, you can do things like you train one odor to be good and one odor to be punished or something, and then watch. So that is kind of the goal. Right now, we're just asking even like, is this an experimental paradigm that we can keep in the lab to study? Why? Why aren't there so many other things? As I will show you in the rest of the talk, almost all our experiments are done in head-restrained animals with the odor being thrown at the nose. Because it's convenient, we can control it very well, but that doesn't seem very natural, right, for the behavior. It's okay for the kind of things I'm going to tell you about, I hope, but if you really want to understand what do they kind of naturally do, I feel like we should be doing this. Simple questions like mice, how much do they actually use stuck chemicals versus airborne chemicals? We don't know that, right? This is the funny thing about mouse. There's so many neurobiological studies in mice, but if you ask, like, what are the natural behaviors that mice do in the wild, most people would say, yeah, go find food, but we don't know. What are they? Are they hugging the walls in real life? Are they going in foliage? So this is semi-naturalistic. I'm not saying this is, but that's sort of the goal. And so then we can do the kinds of experiments you're asking one could do. Anyway, those dots that we're doing, so that was our first encounter into quote-unquote machine learning. So actually there's a postdoc in my lab independently with somebody else, developed actually a deep neural net which can take a whole bunch of training images where you mark, let's say, body parts. You have a bunch of videos, you mark, let's say, the eyes, the ears in, let's say, 500 frames, randomly selected frames. Then you can train it deep in a convolutional net to then play any video and it'll automatically pick all of these things. It's just a very quick trick to pick the parts. And this is now an incredibly well-used tool. It's called Deep Lab Cut. I mean, it's become very big and very friendly usage for people doing behavior. So much so that this is my most cited paper in the last 5 years, even though it's completely silly, right? So, but there's hunger for methods, basically, right? Sort of automatic methods to deal with big data.
You're predicting that part.
No, it's not. That's what it looks like. So we did the video and we're plotting it for our purpose. Yeah, so that would be great, right? So maybe that's the next step. Do a deep network to predict where the animal is going to be. No, this is for visualization to show that that's where it's going to go. Okay, again, I'm not going to talk about it. So let me get into the real introduction here, so to speak, which is that what the olfactory world here, if you're a mouse sitting there, the world is basically a bunch of chemicals that are volatilized and floating around. So the animal, in this case, let's say a mouse, will have to take this collection of chemicals and inside the head, in the brain, presumably separate this out into things in the world, right? Because it's like in visual field, you look at the scene, there is information in the wavelength, the intensity, but also most importantly, the position, x, y, right? So that's automatically, that really helps you group things, like segment objects. Mostly it's about proximity and belonging. But in smells, like in this room, if I have coffee smell, somebody's cologne, and then maybe some other bad smell, you have all of these things. How do you take those chemicals that belong to one, like the apple or the thing, and that's one thing, and there's another, rather than saying all of them together is now a completely new smell, right? Every time you go, that's not what you do. You are able to pick individual things out. And so I'm not sure if this is the way to frame the question, but for me, it seems like an intuitive way to think about collection of chemicals. How do I now essentially segment this into different things? Because I'm emphasizing that because in olfaction, in biology, we spend a lot of time worrying about detection. We say, oh, can I detect that chemical? Can I detect, is this receptor center, right? True, and that's an important question, and we too spend the time. But you never get chemicals in isolation. It's always collection, and then you have to now make sense of that crazy chemical world, and that seems like a very interesting problem. So I put some things, again, what we think, right? I don't know if this is right, but it seems like you have to detect it, but then you have to segment this smell landscape, and then I don't know if these things, two things have to be done together. Maybe all these three things are done in one thing, or it's done sequentially. And then, of course, the whole point of this is to then use this to do something, right? The first problem we encounter in doing this is how do we order the sensory world, right? In audition and hearing, you have a very beautiful, the world has many different frequencies of sound. Your cochlea, your sound system, is very sensitive to specific, your cochlea has sort of selectivity to a high frequency to low frequency. So there is kind of some way of ordering the sounds. You can make chords, of course, but that's a way. Vision, you have color, that's also relatively smooth world, at least the world color, not your perceptual color. Also, there are physical features, like edges. I mean, that's an actual feature of the world that seems orderly because you can change the angle gradually, and there's an axis you can draw. What's the axis for here? Not clear. So I think you have a bunch of chemicals, and how do I change? One obvious thing is concentration. So given a chemical, you can plot that, but that's basically all we have so far. So is the entire chemical world a lookup table, meaning that it's just everything or is there some actual sense in which there's an axis? And again, there's a lot of people thinking about it, and there's no satisfactory answer for this thing. And I want to just,
Just as for taste,
there
are regions which you group it roughly into
Correct.
salty.
Correct. Taste is very much quantized into a few primaries.
There's no analogous.
There are many claims and there are many graves of people who try to, so it's really very unsatisfying. The best we can do is that there seems to be certainly something like pleasant, unpleasant. I think is, but that seems very boring if that's all the fun. It's a starting point, but even there, it's not clear whether there's, everybody who's pleasant looks the same chemically. It's not the case also.
Some species may find something.
Completely. And our own species, different people may like, who can before I went to the US, I didn't even like regular cheese, leave alone blue cheese. Blue cheese smells horrible, but now I actually really like it. And it's mostly smell anyway, right? So that's an interesting question, and I don't know if this field is worth pursuing. There are many people who think. One of my former colleagues, Alain Aspero-Guzik, a chemist, actually a wonderful kind of a quantum chemist, who unfortunately got recruited by the Toronto Mafia with the machine learning and chemistry and all that, Vector Institute and whatnot. But he actually had this beautiful paper a few years ago where they said, okay, the chemicals are structures discrete, right? I mean, you have all these discrete things. Is there a way to make this continuous? And then maybe that way you can kind of order it. And for them, that's for chemical purposes, to develop better chemicals of certain type, right? So you put it, I think, I don't know this enough, many of you probably know this more than I do, this variational autoencoder. So autoencoders are where you take a bunch of stimuli and you keep reducing kind of the representation, right? You have, let's say, 100,000 elements here, they come down to maybe 1,000 elements, you compress it down, and then you send that's the encoding, and then the encoder then is put through a decoder. So the whole learning of these weights across these different layers is kind of minimizing the error of what you predict here for each of these inputs. Right? I mean, this is the standard backprop, the standard deep neural net is just doing classification, categorization here and here. Here is basically identifying one and the other. From what I understand, the variational part of it is to make this latent space more smooth. So instead of having one point, each particular input will have actually a kind of a fuzzy cluster of things in the middle, which apparently imposes some sort of a continuity. But they use this for chemical space by using some sort of an encoding. This is apparently a well-known chemical thing called SMILES, where you can take any chemical and represent it in a linear way. There's some sort of a linear descriptor that gives for, you send this linear descriptor and predict it. One thing they did differently, which is the clever part, was that let's say that you want to make a better organic LED emitter, right? You have a whole bunch of chemicals that do good OLEDs. You want to now make a different one, right? So he defines an objective function here, reading off of the latent space, which then gives some sort of like this is better than the other. With the known ones, you have like I'm here, and they want to climb up or down the gradient either way. So kind of have another, in addition to just reproducing this well, but there's apparently another thing that also needs to be satisfied, which is that you make this as good as possible, as bright as. So for OLEDs, you can use this. I thought for smells, for us, we can use maybe smell quality, like how pleasant it is. So you put through thousands of odors, and here you can maybe do some sort of a pleasant indicator. None of this came to pass because he left. And also, how do you get data for perception, right? What's the high throughput? For chemists, do thousands and millions of chemicals at once for everything. Whereas for human experiments with olfaction, we are very, very data limited right now. But of course, companies like Google are not. So what I understand is Google, actually some group in Kendall Square in Boston, they are collaborating with the fragrance companies, which have a huge amount of data of a chemical and sort of a bunch of descriptors by smell experts from their company, right? Like, it's woody, citrusy, whatever, right? So let's say you have 200 descriptives of each chemical, and apparently they have essentially a collection of millions of these chemicals too, right? That's getting to be big data, and apparently they're now working on something like this to get. But what I wanted to mention this for is that this problem of how do you make this sensible in one axis, maybe a chemical problem, I don't know. So that's where we are. So it makes it hard to study, and we're not bothering in that direction. So I'm not going into the direction to say like, what are the similar chemicals, what are the dissimilar chemicals? We're not really doing that. So instead, what do we do is that we just look at the neurobiological kind of signals that we get when an animal is doing a task, a well-defined task, right? The hope is that by matching what the animal does behaviorally with kind of our inputs and the neural recordings, maybe we learn something about how this is done. And again, you have these chemicals, some task, maybe there's something to be. And again, it's wishful thinking. Again, all of neuroscience, we have techniques. We hope that these kinds of directions will make a little bit of an advance. So smells are detected by receptors in your nose. And many of these objects that we say, right, this is a very Western thing, right? It's raspberry, pears, bananas, pineapples, peach. Each of them, there are a few chemicals that are emitted by those fruits that are characteristic of that fruit, right? Of course, each fruit emits many, many different chemicals, but there's a small number, and I think in this particular drawing taken from here, there are probably a specific one that seems to be the dominant. I don't think they would smell exactly like that, but it kind of gives you the flavor of that. Just to tell you that, that's really what when we mean smells, it's really the chemicals that you are then interpreting. So this drawing may or may not be easy to understand for a non-biologist, but this is a drawing of a mouse's head. This is the nose. They take in the air here. Most of the air is going into the lungs because that's the point. Some of it gets diverted to the nasal epithelium where there's a little bit of tissue here, which is actually for mice, it's a lot of tissue, which are the neurons that detect the chemicals, right? For humans, most of our nasal epithelium is just the respiratory stuff. There's no neurons there. Some part of it is neurons for us. But for mice and other ones which smell very well, there's a lot of this. So this is the main olfactory epithelium where the neurons that sense the chemicals sit. They then send information to something called the main olfactory bulb, and then there's a whole bunch of other brain circuits. This drawing is there to show that there are also other chemical sensors in the mouse's head, right? We're not going to bother with it, but it's not just the nasal epithelium, there's other kinds of sensors. This is a drawing of a fruit fly, which is another big model system in neuroscience. This is the eye, and then this is sort of some proboscis, and then there's something called the antennae, right? As you said, they now sense chemicals. And then they go to the neural circuits, which are drawn here, okay? This drawing, it's too small here, but this fruit fly that I showed you has about 120 different receptors. So the different types of sensors for chemicals, the main point of this figure is that even in insects, and particularly in insects, there's a large number of them. To contrast this, for vision, typically most animals have 2, 3, or 4 different types, right? For us, when you're trichromat, you have 3 different colors, like RGB, the 3 different pigments, plus the rods, which are color insensitive. Certain weird kinds of shrimp have up to 10 or 12 different photoreceptors, but it's still very small. Whereas the olfactory system, somehow we have a huge number of receptors. And this is true for almost all animals, right? For mammals, actually for vertebrates, sorry, this is a different kind of drawing. This is taking the gene sequences of all the receptors, and this dendrogram shows you how related they are. So there's a whole class of receptors that are kind of related to each other, but this is taken from mouse, human, fish, frogs, and so on. So humans have about 350 to 400. Mice have about 1,000. Elephants have, I think, about 1,500 or something, but that's the range of the number of types of receptors, right? They have. So that's basically large numbers, and then the question is why, and we don't know, but one idea is that each of these receptors, so this is one particular receptor in a fly, if you now give 110 different odorants to this, which John Carlson and Eliza Hallam did, and they're plotting the firing rate of the neuron, right? This is the neuron that's sensing, that's OR82. They're throwing different chemicals at it, right? They have reordered it in a funny way where they put the most sensitive, highest firing in the middle, and then they slowly kind of put the rank order the other one. Somebody asked me when I gave a talk to other physicists, like, why is this symmetric? This makes no sense, right? Because it looks like a tuning curve. But what really is that you should have just rank ordered it from the highest to the lowest, right? The symmetric thing is just to sort of show you how narrow or broad it is, okay? It's made up. This receptor seems to really like one chemical and most of the chemicals not that much. This receptor seems to like a lot of chemicals almost smoothly, right? If you go for the negative means the resting firing rate is suppressed. So these neurons typically have a resting action potential rate. So that is reduced. And that turns out to be actually quite common in insects. We are now finding that it's also probably true in mammalian neurons. I mean, it sort of makes sense, right? I mean, if you have a little bit of an idling, maybe you can increase your coding capacity by maybe. So this is now from they looked at, there are about 80 different receptors. They looked at all of them in fly, and there's basically zoo of tuning. So something that's very narrow to rather broad. And the idea of this is that if the universe of chemicals is large, people are trying to estimate, like a given animal, how many chemicals in the world will an animal detect? And the sad thing is that we don't even know how to begin to estimate that because you need some statistics of responses and whatnot. But it's certainly in the order of, it's certainly greater than 10 to 100,000. Because if we randomly pick small molecules from a sigma catalog and throw it for mouse neurons, they will more or less fire. Somebody will fire for that, right? So you can almost sense any small chemical that reaches your nose.
I think that's not necessarily detection, right?
No. That's right. So this is basically, exactly, you're just looking at, but what do you mean, but not by the animal's detection? Yeah, correct. Yeah, we're just talking about the firing of the neuron itself. And then what happens downstream is a good question, yes. Good point, yeah.
So there are 1,000 receptors in a mouse.
Mouse, yes.
So, but then, so how do they detect 100,000 smells?
Well, we don't know the answer, but I think if you think of it's a combination, yeah. That's, I mean, we can, the actual, part of my punchline of it's not a punchline, part of the thing of the talk is we did an experiment where we can show that for most everyday things we can think of, simple linear classification is good enough because you have a lot of receptors. Each of the receptors only responding to a subset of the universe of things. So let's say 10% of that. Then you have enough zeros so that you can actually figure out what the right combinations. But you need the population. But whether you need the whole population or can you sample some subset of them, those are all questions we don't know the answer to, yeah.
The olfactory ability of an animal is directly proportional to the number of receptors?
Right. So again, it seems reasonable, but again, it's hard to know what experiment can you do, right? There are at least two parameters. One, the number of different types of receptors, so that difference, different discrimination. There's also the copy number, right? Like your 10-pixel camera versus 20-pixel camera versus 30-pixel camera will maybe have better sensitivity. So those are, we think there will probably be different things where you have more detectors, maybe more sensitive, more types, maybe more discriminating. So we don't know, and the main problem is it's not that we don't know, it's just we don't know how to exactly test that, right? What does discrimination mean? How do you test? I think one good experiment could be that there are so many breeds of dogs, right? They all have very approximately first order similar genome. You can then go and look at all their different receptors, look at the ability, look at the copy number, because the small dog might have only a few, a bloodhound has a lot of. So I think you want to take one narrow species with variance along one direction, like more types or more, something like that, yeah. So it seems reasonable, but we don't know if that's the case. Okay, so the other point is natural odors are blends, as I said. So take a banana, and then if you do gas chromatography mass spectrometry, so this now in this axis is something that tells you the different chemicals, right? So you see that a banana, if you volatilize it, has many, many, many components. Some components at very high concentration, some at low concentration, but it's misleading to think that the highest concentrations are the right things. Because it may turn out that that particular animal does not have a sensor for that. So this is actually an annoying problem because you can't just take an object, look at the chemical spectrum and assume that it's okay. You have to then multiply or convolve this with the actual sensitivity of the receptor thing, which is not easy to measure, right? So, but for flies, people have measured it, but here. So anyway, so those are some names. The one thing that's pretty clear is that it has to be volatile. So the thinking is, I think it's in around 200 Dalton, like some molecular weight is what can make, which is why plastic, like in a polymer, is not going to smell. It's only the things that break off, right? And the smaller parts that, smaller molecules that come off that you can smell. But within that, large number of things can be smelled. Fruits typically have a lot of these esters, right? The COO, right? I mean, RCO. So they have a lot of these esters. They have some alcohols, but they become ripe and they become esterified. So those are the things that we tend to use a lot in the lab because they're relatively pleasant smelling, and there's a huge diversity of them in real life too. Okay, that you have seen this. So the segmentation, just to carry it further, if the visual scene, right, if you have this thing, if I say, where's the basket of jasmine, there's multiple, I mean, you're effortlessly doing it. As soon as the image is flashed, you've segmented it. But if I say, okay, here's a collection of chemicals, what's the jasmine here, right? That means that you have to kind of figure out those that belong to that and then somehow group them in your head, right? So this, then we asked, so that's the motivating question. So now the experiment we actually did was to say, okay, if I give a collection of chemicals, can a mouse detect a particular, I say like, oh, I have vanilla smell in this mixture, can you detect it, right? So that's in some sense an approximation of the segmentation, right? You have all these distractors, can you pull out the thing that you're asking to detect? We don't use complex chemicals because it's hard to reproduce from experiment to experiment, so we use pure chemicals. So we mix a bunch of pure chemicals, and ask the animal to detect, train the animal to detect one of them. Let's say that one, right? So here's the behavioral experiment. We picked 16 different odors semi-arbitrarily, meaning based on history, what other people have done, what we have done, but also trying to say, okay, here are some chemicals which we're going to say they're similar because the molecular structure kind of looks similar, and here are some other dissimilar ones, right? If you ask me, put me against the wall and say, what do you mean exactly similar to similar? I can't really defend, but it's like a bunch of esters that look similar, sort of alkaline, alkyl chains, and the other ones have like weird benzene rings and so on. Okay, that's what we did. So if you now take 16, there's a large number of mixtures you can make, with just binomial model, but we subsample this and we use in the order of about 16,000 mixtures just for practical reasons. So the experiment is that in each trial, we will give some subset of these chemicals, let's say this, and this, these four, and the animal will tell us, yes, this is the correct trial if this odor is present in this mixture, independent of anything else. In the NoGo trial, what we call, if that particular target is missing, the animal should respond differently. What we're asking for in this animal is to lick a water port if it's the correct answer, if the target is present. And if the target is not present, they have to sit still and not do anything. So it's a go and no go. The reason they're licking is that we're water restricting them in the cage, so they're thirsty. So this is what they do in the lab, they now get water. Right? That's why we train them. The behavioral shaping happens over a few days. The mice are actually very good at learning olfactory tasks, which is good for us, but that's the thing. And so different trials will have different mixtures. Right? So that's, I hope, the actual task. By the way, the grid, other people got confused with this grid. It's just my way of visualizing which chemical is there in that mixture, okay? I could have drawn it as a linear vector, but I just drew it as a grid, okay? So here's the video of the animal. So it's going to play and repeat. So this is the odor port. So we have a device that does all these different mixtures and delivers them through this tube with the air. There's constant airflow, so the pressure changes are minimal to non-existent. It's just the odor comes on or off. This is the lick port, which gives them water. They're head restrained because we want to do other experiments neurobiologically later on, so this makes it easier. And the trials are, so let's wait for the trial. So that's a no-go, meaning that mixture did not contain the target. This animal shouldn't lick, and it didn't, because this is a well-trained animal by now, right? So you can see he's sniffing, right? That was also a no-go. No, these are all relatively neutral odors. So in the beginning, these odors have no meaning for the animal. So we are assigning the meaning by, so the behavioral shaping is being worked out not by us, but collectively by the field by a long time, right? This is actually the one good thing psychologists have done for us, so I shouldn't say that, but this behavioral shaping, you can start with simple tasks, and they kind of start associating, something comes and I'm going to get a reward, something. So it takes a few days, but you can do it. Yeah. Okay, so that's basically the task. So the nice thing is that you get this and we can get lots of trials. So we can get about 200 trials each day, and then you can keep doing it over and over and every day. And then the percent correct that the animal does starts off initially at 50% because that's chance, right? Go, no go is 50%. And so each day is about 250 trials. So 2 days, 4 days, 6 days, right? By about 1 week, they are doing about 90% correct in this task, right? And then we can show that, it's still not asymptoting. I think it's going, and they get up to about 95%. The control is that if you scramble all the identities of the thing and just do random things, right? And then to show that they are using the chemicals to do this. It's not some other cue that we have not paid attention to, right? So we can train them to do this task where I give you an arbitrary mixture, and we think it's unpredictable because there's 16,000 choices. So presumably the mouse cannot, memorize all the different choices. So it's looking for the invariant among all of these other varying things. So there's stuff that's nothing to do with what I want to do, and you're honing in on picking out the task, right? So the first thing we did was post hoc. So the actual trials are given completely randomly, pseudo-randomly, because we do random number generator. Post hoc, you can sort all the trials with only one chemical, meaning it's either the target odor or not the target odor, right? There were no other distracting things. So that's an easy task, right? And they would basically do it around 95%. You had a question? No, it's better now, yeah. So they're doing it, and this is where 90% of the olfactory scientists, the field, that's where we people study. They just do simple odors, give yes, no, is this or that, and it's fine, but the point is that it's extremely easy, and my analogy is that if I put you in a room and switch the lights on or off and ask you to tell, I mean, that's one bit, so it's really, really simple, right? I think for mice, this is really simple. But for us, the shocking thing at the time was, as you increase the number of distractors, even up to 14, they're still extremely good. So this is average across all animals, all combinations, right? And so I think this was our first surprise, because our hope was they will sort of dramatically fail, so they would be good, and then they will crash to 50%. And that kind of form of crash we thought will tell us something useful. Because when things are working, it's hard to know what's going on. But anytime there's something interesting changing, but unfortunately it's happening super gradually here, right? An aside, which this is not published and probably not publishable, it's kind of amateur experiments we did in humans, where we, with collaboration with the psychophysics, visual psychophysics person, we just tried the same experiment in humans. We put them there and gave these odors. And these humans are, of course, people in my lab. So we are terrible. At least untrained humans sitting in the lab are actually don't seem to be that good. I am pretty sure that more trained humans and better experimenters than us, better human experimenters than us, it's probably going to be better, but I don't think it's going to be as good as this. What this means is, as for us, when you start mixing more and more chemicals, we kind of lose track of like one particular thing in there. And I think, our own experience is probably true. Yes, if you really obsess about coffee, right? I mean, like me, you could probably pick up the coffee even at low, but many other odors, it's kind of hard once you start giving mixtures. Anyway, this is just an aside.
When you give mixtures, it's sort of really distributed.
So this is a good question. So we don't control for concentration right now. So each mixture, we are using 1% of weight in the solvent, which is mineral oil. When they actually come out, in terms of the actual vapor pressure and the number of, the sort of the molar thing, it's going to vary widely. We didn't control for it because what we said is it's hard to control. And what we know is that if we go, and let me just go back, this is an important point. Oh, here actually. If it turns out that this odor is just really high vapor pressure, so it's really a lot of the molecules and therefore it's easy, then if I use this odor as a target and then give this as a distractor, then this should be much worse, right? We have trained different mice with all of these different chemicals at the same kind of levels, and we don't see any difference. We can train. So yes, there is different effective concentration, but they're all in the detectable range. But this brings up the question, which the experiment which we haven't done, which we'd like to do, keep everything the same, but then gradually titrate this chemical's concentration lower and lower until you start seeing the failure, and that would be a good experiment to do. Yeah. The proper way, the people who do human psychophysics, the proper way to do this is for each chemical, for each subject, you find the threshold, and then you do multiples of the threshold, right? But that's a hard experiment, and for mice, how do I ask the mice to tell me a threshold? Then you have to train, so it becomes very complicated. But for humans, people do that. You take every chemical, you titrate the concentration until you detect, then you mark that as your threshold, and then you do everything multiples of that threshold. Okay, so, but then, sorry.
Hang on a second, is number 14,
Mm-hmm.
is that one go odor with 13 no?
Correct, that's exactly right. Ooh, yes, you're exactly right, yes. Yeah. Well, it's not exactly, it's a target odor. It's like, right? Go odor means it's a mixture, but yes, I think you mean exactly the same thing. It's that what the odor that we are asking it to look for mixed with other things. There's only 14 because we chose 2 of them as targets, which I didn't. I mean, it's a little bit of a complication. For convenience, we did that, which is why we didn't go up to 16, right? Could have been, okay? So, so what can we do with this now? This is fine, but the point here is that let's say you take this point in the axis 10, but this mixture of 10 is heterogeneous mixture, right? There's a mixture of certain 10, another 10, so I'm mixing them all up and adding. But what if there's a way to separate all of this out, right? It's a very complicated, problem. We don't have enough data to do it, exhaustively. One way which we can do is that we actually pick this somewhat cleverly, well, semi-cleverly. that all of these odors are these so-called tiglates. So they are particularly the CH3, you can sort of see CH3, CH3, COOA. So there's this group that's it's a particular ester that's fruity, and then there's all this other. So we're going to call them similar, right? Just because they look like that. And these are sort of dissimilar odors, if you will, right? So if I have this odor as the target, will the mouse perform worse when these guys are the background distractors versus if these guys? And the answer is yes. So here's a plot where each of these pixels is the performance. 100% correct is perfect performance, 50% is chance. As you vary, it's the two variables. One is the number of non-tiglates, so these guys as you go, and this is the number of tiglates. And what you can, I think it's a little bit hard to see in the contrast, most of the variance here is going in this direction, right? If you march in this direction for each of the columns, the colors look about the same, right? You can, of course, quantify that and say, like, if you now plot each of these lines here, right? And they look basically almost the same. So the color here is the number of non-tiglates in the mixture. So you fix the number of non-tiglates. Let's say you pick one non-tiglate, and then you change the number of tiglates. So you have one dissimilar stuff as your background, and then you slowly titrate up the number of similar ones here, and it marches down. Now you take two non-tiglates and march, right? All of them more or less lie on the same. These are, of course, the regression lines, not the original data, right? Just for visualization. But if you put the margins in the other side, it basically is more stepwise, meaning if you have, let's say, two tiglates, as you change the number of non-tiglates, your performance doesn't change because that presence of the two tiglate has done all the job. The number of non-tiglates is now an irrelevant variable. So this is basically going down like. So it's somewhat satisfying, except for the fact that why is this similar, right? It's this kind of chicken and egg, right? We say, oh, we saw this and therefore we believe it's similar. But if this hadn't panned out, we would not be sure that it's similar and dissimilar. And I think this is a big problem in olfaction, goes back to this axis. We have no axis where we can say, 1 is next to 2, 2 is next to 3. There's nothing like that. Similarity, for the most part, is in our perception. And for humans, you can ask them, you can give them 2 and say, like, is this similar or dissimilar? And those kinds of experiments, people have done or trying to do more. But for mice, to say what is similar and dissimilar is much more complicated. And in fact, given that our experiments work like this, we are wondering whether this experiment could be a way to define on a similarity. But anyway, so there's just, essentially this is behavioral, right? We're just saying, here's some chemicals which I'm doing something with behavior. But given what we know about the receptors, and given what we know about a little bit about the circuit, is this task hard, easy? Is it like, logically, can we understand it in some way? And so to do that, so let me tell you a little bit about the circuits in the mouse and other animal brains. Yeah? Yeah.
Or it's sensitive to things which are.
Yeah. So, yeah, it's not clear. The preponderance of evidence seems to be some version of shape and sort of surfaceness, if you will, right? But there is, I think, maybe you're alluding to theory, there is this idea that I don't even know where it first came up, but this guy, Luca Turin, from a physiologist, made popular the sort of vibrational theory. So the idea is molecules can go and the receptors can essentially do Raman spectroscopy in the thing. And there's not too much evidence for that, but it's not that it's completely ruled out. It's a good question. Empirically, all we know is that what the tuning curves that I show, if you throw a whole bunch of chemicals, you get some sensitivity measure, but to go back and test what you do, you have to do much more systematically by varying things more precisely, and it's not clear. I think most of us, I don't know, for some reason or the other, for most of us, it seems like the general shape seems to be the more simple explanation, but doesn't have to be the right explanation. Yeah. Okay, so this is the olfactory epithelium where the receptors are there, they detect the chemicals. The axons take the spike signals to the olfactory bulb. There's a bunch of processing that goes on, neural processing, and those connections then go to the ventral surface of the brain, which is below here, and that's the olfactory cortex. Right? So this is kind of the simple, and then there's 2 sides and there's cross projections and so on. So in terms of the basics, I said there are 1,000 types of sensors, and there's about 400 in humans. The one key thing is that unlike in many other sensory systems, there is no kind of nearest neighbor topography. So in the visual system, it makes sense, right? I mean, your space, the retina has your spatial map, and that spatial proximity is preserved neurally all the way. So if you go in the visual cortex, neurons, neighboring neurons will respond to neighboring points in space. The auditory system, neighboring neurons will typically respond to neighboring frequencies, at least in the tonotopic maps, right? So, and there are computational maps. For example, there's maps of location, which is not a physical thing, it's kind of a computed, a map. But in the olfactory system, we can't really recognize any, and that makes sense, right? Because if the chemicals are kind of no similarity, no kind of way to order, but that's what it is, it's a fact. Yeah?
There are 1,000
Yeah.
mice.
Mm-hmm.
Would that be a reason for the failure in your
I don't know, you tell me. So quantitatively, I mean, to me, there's two ways to think about it. To me, it seems like it's in the same order of magnitude. So why should it be that different?
Well, evolutionarily, we have lost the sense of smell much.
But why?
Because most of them have become pseudogenes.
Sure. But we still have 400, right? I mean, 400 is just 2.5 times smaller than 1,000, right?
But still, if the panel on the left side, you know,
Yeah.
the chemicals that are sort of similar,
Yeah.
which is bi-structured,
Yeah. No, you're right. So I think your question really is there, but can we formalize it and show in some mathematical way, right? I think it is a good question, and that's what I mean. I think I don't know the answer, but it would be cool to think about that. Yeah.
Maybe instead of 400 and 1,000, the right comparison is roughly 2 to the 400 versus 2 to the
1,000. Ah, good point. Okay. Yep. Yep. Yeah. Could be. Good. Yeah. Having said that, also, there are people like wine tasters and coffee tasters who do pretty well, right? And I think it would be good to compare because our mice are like expert coffee tasters, right? Because we have trained them whereas the humans were like idiot humans. Anyway, that's a good point, actually. It's not really the number itself, but yeah. So that's one thing. And then this thing, which actually has gotten a lot of attention from, many theoretical neuroscientists like Sampolinsky and Abbott, that these projections from here to here seem to be very sparse and disordered. So it's kind of like a random matrix projections, and people are now making nice theories about it. So this is a beautiful slide from Vijay Balasubramaniam's preprint, which is still not published, where they say, okay, there's a space of chemicals, which is, I think you were sort of alluding to this, a whole bunch of chemicals. A given natural odor is some sparse sampling of this. So a small vector with lots of zeros, a few ones, right? I mean, this chemical. So we don't know what exactly this is, but it makes sense heuristically, right? If I'm sensitive to 100,000, not all of them are there in this moment. It's only a very small subset of them. And then you have the receptors, the olfactory receptors, which are now sensing these. Each of them have this kind of a tuning curve that we said, right? So there's the actual smells present. They're sensed by this array of receptors. Then in the olfactory bulb, there's stuff that goes on, and some ideas are that you want to change because there are all kinds of different levels of sparsity. Maybe you want to kind of equalize it so that every smell has some relatively similar representation. At least this is the thinking. And then maybe you want to decorrelate things. You don't want to over-redundantly represent. Very vague right now, but there's a bunch of circuitry in the olfactory bulb that needs to be understood. Okay? From the olfactory bulb, this is this sparse random projection. So a given mitral cell, the output cell of the olfactory bulb, so the receptor, mitral cell, you can think of it as kind of belonging to the same thing. That mitral cell, that channel, now projects to several cortical neurons, but a very small fraction, something like 10% of the entire population. And as I said, there's no known topography, right? So this is some weird weight matrix here going from here to here. And the idea is that this kind of projection helps you flexibly now and quickly, kind of categorize whatever way you need to. If I tell the mouse, look, you got to take these chemicals and separate them from these chemicals, having this kind of a projection perhaps makes this kind of a downstream separation easy. But the problem is that I don't think we can really defend it. There are many other ways to do the same thing. The short answer is we don't know why this kind of projection is there, and I think people are kind of thinking about why this is the case.
Full of memory here.
Mm-hmm.
I mean, if I close
Yeah.
my eyes and I smell gas,
Yeah.
I remember I
Yeah.
smelled it sometime ago.
Sure. Yeah.
Why is it so?
Yeah, it's all feared. Yeah, it's because it's very easy and very simple. So it's a very good point, and I think all along the way there are backward projections. Okay, so there's projections from the cortex to back to the bulb. In fact, we're studying this just empirically. We don't have hypotheses. We're just trying to understand what these things are. Like you said, the cortex gets projections from these higher cognitive areas, which presumably bring memory and whatnot. Absolutely, there's a lot of recurrence. It is not a feed-forward circuit. We're just drawing this for convenience, right? I will also say, though, the memory, right, that's exactly true, and this seems to be cross-cultural, right? I mean, almost every culture's literature brings this up, right? But the thing is that the odors evoke memories, but the memories themselves are not usually about the odors. They're usually about circumstances and situations and scenes that you did.
I was talking about just recognition of odor.
Right. Oh, yes. Correct. Absolutely. But that I think can be explained by this, because if you have these things and you've now made this weight matrix such that you separate things out, the next time a Jasmine-like thing comes, then you're going to go into that state. So you're putting similarity matching there. So, yeah. Right, that's right. And that I think is one of the motivations for doing this. Yeah. Yeah. And that assumes that this matrix can be constantly learned, and there's actually good evidence for that. Yeah. Okay, so I have a few minutes. I think I'm just going to go through this, the first part. Actually, I'm going to skip this part, and I'm going to go to the second part just because you prompted me on this, because I think this projection is actually kind of interesting, because this is right at the edge of something non-trivial, but also experimentally tractable. So it's not so crazy that you think about stuff and nothing happens. So I'm going to go to that. The part that I'm going to skip several slides basically is looking at the detection of these, like what is the coding of this. And I'll quickly tell you what we found is this, that not we found, we confirmed what other people found. So if you look at basically a bunch of odors, in this case we used about 35, and look at in one experiment, let's say 50 sensory neurons, right, of different types in this case. The size of the circle is how responsive they are. And this just shows you, look, certain odors activate many neurons, other odors activate none. A given receptor, a given neuron has some sort of a tuning curve across this. So it's this population that's giving you kind of the overall picture of each of these things, right? And this actually is an interesting. The horizontal axis is different odors. So we just randomly picked about 35 odors. Yeah. That's right. It's just some labeling of that. Yeah. There's no. 15 next to 14 means nothing. So this one is interesting because it's a paper by Yuhai too, which kind of replotted some of these published data to show that the affinity of odor and receptor seems to follow some sort of a log normal distribution. And the main idea is that there's going to be a long tail. So there's a few things that are really sensitive and a whole bunch of stuff on this stuff. I don't know if it means anything, but that seems like what it is. So I was going to show you a little bit about mixtures to show that the mixtures interact actually in complicated ways, unfortunately. Meaning that if I have one odor that activates, another odor that activates, giving them both together, it turns out to be a nonlinear. Typically, there's antagonistic interactions. That turns out to be the most prevalent interaction. And with Massimo, actually, this is what Gautam Reddy and Massimo, we had this nice sort of a biophysical theory, if you will, for why you might get those kinds of interactions. And why that interaction should be prevalent, is it just an accident or whether it's useful for representation, unclear. But to go back to one main thing which I think you were kind of alluding to the population, it turns out that this task, right, detecting this odor independent of what else is there, right? You can pose it as like, can I make a linear classifier that takes my mixture response here and then says go or no go, 0 or 1, based on whether it belonged to this column on the right. So all the neural patterns corresponding to my go trials, are they linearly separable in a learned way from all the patterns for that, right? And the answer was a resounding yes. So it turns out that we can vary the trial-to-trial response of the neural response that we measure up to 100%, and then still get in a linear classification after you've trained it, pretty good. In the range of the variability that we actually see in the mouse neurons, the linear classifier can give you very, very good performance. So this entire task, I think it just simply is because the sparseness of the activation. A given odor is activating a sparse population, and if there's enough kind of a non-overlap, you're going to be able to determine that. And we can also reproduce this idea that the more distractors that you have in your mixture, our linear classifier as well as a nonlinear SVM, support vector machine, can kind of do this. So at least formally, this task, given your receptor activation pattern, you can learn downstream to do it. Of course, we're not saying mice are linear classifiers, but the task formally is not a hard task, right? So then why do humans fail, right? Going back to the thing, is it right? And I think 400 is also kind of enough if you really look at the glomerular pattern. So it's something more. There's some other constraint that we don't, we're not kind of taking into account, okay? But I think for 2 minutes, what I want to tell you is more or less pose a problem, right? Which wasn't even thought of as a problem, but suddenly we realize it's a problem, which is that this is the understanding, right? The projection from the olfactory bulb to the cortex, where we think all the fancy stuff is happening, this is essentially a sparse random projection. That's the hypothesis and thinking right now in the field, right? Mice like us have two sides, left and the right. So you have, let's say the left olfactory bulb is projecting to the cortex. Yeah. Okay, random is the word they use. So the point is that if I take a neuron from here, coming from one channel, and look at the axon and where all it goes, right? Look at another neuron that belongs to the same glomerulus, right? Look at where it actually, those two things seem to be uncorrelated, right? Of course, I thought what you were getting at is random is a loaded word. What does that mean? So they haven't seen any structure so far. So people are calling it random, right? I think the real thing is that it's sparse again is a loaded word. A given input only projects to 10% of the neurons that are present. That's known. But it's not like the input is localized here, right? It's kind of spread out. So it's disperse and sparse, right?
Many neurons can do the same job.
That's the question. That's the question. So I think all the observations right now are just what I just said. The anatomical projections are all that's known right now. Everything else needs to be. So I will tell you one example, essentially saying that it cannot be random. It is sparse and dispersed, but there is micro order. I mean, it almost has to be, right? And I think we have a clever way of showing that why there's, because it's a hard thing to show, any other way. So the logic was simply that each cortex not only gets the ipsilateral, so my left nostril goes to the left olfactory bulb, goes to the left olfactory cortex, but there are, in the brain, as you know, there's cross projections. So through a crossing of the hemisphere, the contralateral cortex will project to this cortex. That projection also has no known order, right? So you have one random matrix, and effectively you have another random matrix coming from the other side. So if you have a set of neurons, if it's truly random, this and this, the neurons here will give you different response patterns to stimulation on this side and this side, right? If it's truly random, because right, that's what you would expect from this random, right? You can see where it's going. So we designed an experiment to stimulate selectively one or the other nostril, record individual neurons in one side of the cortex with 15 odors. I mean, this is the method, and this is what we found. Hardly random. So these are example neurons where we're plotting the firing rate above the baseline. So negative means that they got inhibited from the baseline. This is the ipsilateral response, meaning I gave 15 odors in the ipsilateral nostril, recorded the neuron. Contra means that it comes from the other side, goes to this cortex and crosses over and then activates this neuron, right? Those responses are very correlated. And of course, I'm picking some good examples here. But if you look at the overall population, the percent of cells that are correlated in the first cortical area is extremely high. These are 3 mice. I think this is just a statistical thing. All of them are significantly better than shuffle controls, which are not shown here. But something like 40 to 50% of the neurons are. And this is even more striking, where actually, let me back up here. That this is now, for one odor, all the neurons that we recorded in this region of the brain, so each point is one neuron, its response to that odor from the left or the right If a neuron responds really well to that odor, it'll respond well to IPSI and the contra, both, right? So, and in collaboration with Cengiz Pehlevan, our theorist, we basically showed that you can't get, you guys will get this very easily, that this kind of a correlation cannot arise by chance, but how can it arise? And the collaboration with them was to show that simple Hebbian learning, this kind of a correlation-based adjustment of the weights, meaning that every time you have one odor here, you're also going to get the same odor on the other side, so you will kind of get correlated patterns of activity coming from here and here, and that is enough to adjust the weights, and like kind of locally for each neuron, you can get weight. So this is there. I'm happy to talk more about this, to say that, in fact, he even calculated, it's like, how much structure do we need? It has to be 100%, and then we did the calculations to say something like 40% structure, meaning kind of some loose Hebbian correlation is sufficient to give the experimentally observed. The bottom line really is that this is not, if you read the literature, including Larry Abbott, they say, oh, the projections in the bulb of the cortex is random, sparse, whatever. And yes, it's sparse, it's dispersed, but at the micro level, the synaptic weights, they're clearly not random. They have to be highly correlated, which makes sense because I don't think you want to smell one thing from this side and another thing from the other side. You want to probably match that, yeah. Okay, so I'm going to stop there, and I'm happy to, of course, talk more, but I don't need to put the summary up. But I think in the olfactory system, there are many interesting things that, maybe you don't need complicated theories, but sort of to make sense of the observation and to drive what is it that you want to do next. I think there is some room to kind of think a little bit of theory there. Yeah. Thank you for your attention.